Hello everybody, welcome in today's video. I will give you a general introduction about neurodegenerative disease. In this first part we will talk generally in neurodegenerative disease like Alzheimer's disease and Parkinson's disease, and in the next two parts we will focus on the Alzheimer's disease, its pathology, pathophysiology, etc., and the same for the Parkinson's disease. So let's start. So here let's start about some epidemiological study. For example, here in Europe and 13 countries in 2010, I will try to present you, give you some epidemiological studies. For example, for Alzheimer's disease in 2010, Alzheimer's disease and related dementia affect 6.3 million in Europe. For example, Parkinson's disease 1.2 million, for the stroke 1.3, traumatic brain injury 1.2, and multiple sclerosis 0.5. As it mentioned here, all those statistical here as a total give us 10.5 million per 15-14 million, the total population of Europe in 2010, and this effective represents 2.5% of this population. Now, for the psychiatric disorder like anxiety disorder, it affects about 61.3 million in Europe. Psychiatric disorder, addiction, mental, etc., etc., as well as it is shown here, and you can show that the total of all the psychiatric disorder affects about 97.5 million over 14 million, which represent 20% of the total population. Now, coming to the neurodegenerative disease, the main characteristic of the neurodegenerative disease. We can distinguish two types of neurodegenerative disease: acute disease and chronic disease. For the acute disease here, acute brain lesion, it's due to the, for example, ischemic accident or trauma, and it is very often massive. And there is no selective aspect of brain lesion, including neuron and glial cells, and there is a possible at a distance lesion, for example, penumbra area, and it is non-selective character of the dead cell process and necrosis mainly, and it is frequently inflammatory process. Now, coming to the chronic disease. The neural death could be systemic in nature, neuronal selectivity. It can be the cause and mechanism of the neural death can be more and more specific, and the apoptosis is the general process of neural death. Now, for the next slide, the main neurodegenerative disease in the world are Alzheimer's disease and related dementia, and Parkinson's disease, and in this three video, this one of today and the next two, we will, as I already said in the introduction, we will focus on Alzheimer's disease and Parkinson's disease. And now some basic questions related to the neurodegenerative disease. So everybody maybe ask those questions. What about the cause of the neurodegenerative disease? What is the cause? What caused this neurodegenerative disease? What about the physiopathological process? What could be the mechanism of neuronal degeneration? And what are the possibilities to cure the disease? And is it currently possible to stop the evolution of the process? And what is research doing? So we will try to answer all of these questions during this three video, and follow us, and let's start. Now, for the brain lesion, our brain lesion are generally associated to the main disease, and it's a semiology. For example, here the table here summarize some information about Alzheimer's disease, Parkinson, Huntington, etc. For example, Alzheimer's disease, it affects mainly anterior cortex, the hippocampus, which is essential for memory, and as information, Alzheimer's is characterized by the loss of memory, the frontal cortex, which is necessary for the planning and to take a decision, the basal forebrain, the parietal lobe, the occipital lobe, the vision, the amygdala in emotion, locus coeruleus, and rough nucleus in the motor movement. Now, for the phenotype of Alzheimer's disease, the projection nerve multiple transmitter, for example, glutamate, acetylcholine, noradrenaline, and serotonin, and low CBPs are affected in this disease. Now, for Parkinson's disease is generally defined as progressive neurodegenerative disease, and mainly affect the substantia nigra, in which it causes the degeneration of neuron, the dopaminergic neuron, which cause the motor coordination deficit and many other symptoms. It also affects the frontal cortex, the locus coeruleus, and the rough nucleus. And for the phenotype, the projection neuron is primarily dopaminergic neuron and low CBS. Now that are general information. For example, here the schema here represents the brain with the frontal cortex, with the parietal cortex, occipital cortex, and here the cerebellum, etc., etc. And here, as you see here, the substantia nigra is located here in the, and its related cortical structure. The striatum also is located here and its cortical structure. The hippocampus in this region, hippocampus and related cortical limbic structure as amygdala, etc. And we will see all those in the next video, but let's just give a little bit information about that. So now for the histological hallmarks of the man disease. For example, Alzheimer's disease, those two presentation here show a representation of analysis of the brain of a patient that suffer from Alzheimer's disease. So presents, as we see here, that the Alzheimer's disease is characterized by the presence of amyloid peptide aggregated in the extracellular place, or the senile plaque, beta amyloid or senile plaque. In the intracellular space, the neurofibrillary tangle and labeled by the antibodies direct against the hyperphosphorylated protein, and those two main properties are characteristic of the Alzheimer's disease. Now, from the physiological to pathological cleavage of amyloid precursor per protein. So what happens here in the normal case, we have here a gene with extremity COO and the other extremity H3N, and we have here the amyloid precursor protein. We have the beta secretase, alpha secretase, gamma secretase, and those in the normal condition, this gene will be cleavaged by those three enzymes, by gamma secretase here in this part, by the alpha secretase here, and by beta secretase here. And here this one represents the proliferation, the CE regulation, and here the transmembrane regulation. Now, for the extracellular part, as we already said, here these three enzymes are very, very important enzymes in order to generate normal cleavage. Now, coming here, for the amyloid precursor protein, and here amyloid precursor protein is alpha. Now, in the abnormal, there will be here absence of cleavage by beta secretase, and the cleavage is only by the alpha secretase and the gamma secretase, and which label as those two compound here, and those two compound here leads to the accumulation of those two of this one here, because it is not undegradable, which aggregates A beta and form it leads to the increase of CE2 plus neurotoxicity and abnormal size of neurites aggregated here. To summarize, here represent the normal case in which through enzyme cut here, the first one cut here, the second one, the third one, and all is okay here. But when we come to the pathological condition, we have those two enzymes, just two of them are working, and the cut will be in a wrong place, so it will lead to the aggregation of E beta plaque or senile plaque, which are the main characteristic of Alzheimer's disease. I know that it's a little bit complex, but we will see all that in detail in the chapter of Alzheimer's disease. So the amyloid precursor protein, when cut by the incorrect secretase enzyme, can form amyloid beta plaque, as we already said here. Here this one, and accumulate, leading to Alzheimer's disease. And this happens when there is a mutation in the presenilin 1 gene. And for the early mechanism identified by Carla Skatstein, Science 2011, the beta amyloid began destroying synapses before it clamped into plaque that leads to the nerve cell death. And the beta amyloid first forms small clusters that are still soluble and can travel freely in the brain, and then after in this clustered form, it binds strongly to the receptor or nerve cell and initiates an intracellular process that erodes synapses. This synapse, called the PRP in mice and LRB2 in human, is a highly affinity receptor for the soluble cluster form and is located close to the synapses. In PRP expression Alzheimer's mice, the binding of soluble proteins significantly increased activity in an enzyme called coffulin. And coffulin activity is also much higher in Alzheimer's disease brain compared to the healthy patients. And coffulin breaks down actin and building block protein essential to maintain synaptic structural change. I know that I talk a lot in the slide, I say many things, but we will see all this in detail. Now I think I will stop the video here because the time is not sufficient, so I will stop here, and in the next video I will continue. So see you in the next video. Good luck.